Uh, Today we are going to see what is Diauxic growth curve. So as you know, di means two. So this is a growth curve which is having two phases. So first we will see why you get two phases in this particular growth curve. So when you grow bacteria in a flask having limited amount of nutrient, or in a container having limited amount of nutrients, you will find that you get particular pattern of growth which is having lag phase, log phase, stationary phase. And when the nutrients get depleted and the toxic molecules or waste molecules accumulates in that flask, the cells enters in the death phase. So this is a typical pattern that you observe when you have single type of carbon source in your medium. When you grow E. coli like bacteria which is having some inducible enzymes or operon system in it, in presence of two carbon sources like glucose and lactose, you will find that since glucose can be easily utilized since it is a single monosaccharide molecule, E. coli prefers to utilize glucose first, and once the glucose is utilized, E. coli shifts towards utilization of lactose sugar. So when you have two such carbon sources, one is simple and one is complex to degrade, the culture shows typical type of growth phase which is having two lag phases and two exponential phases, one stationary phase and then that phase. So this pattern of growth having two phases in it is called as Diauxic growth curve. So as we have discussed, glucose is a monosaccharide compared to is lactose is a disaccharide made up of glucose and galactose. So that's why E. coli prefers to use glucose first, and once the glucose is utilized, if it has to grow, it has to use the lactose sugar from that medium, and then a new exponential phase starts in that particular broth. So in this diagram you can see you have a first phase shown by orange color of glucose utilization, and the second phase shown by blue color which is of lactose utilization. A straight line initially which is present is the first lag phase, then you get exponential phase which is obtained by utilization of glucose, in which the glucose is utilized and cell growth takes place. At the end of this phase, the glucose is completely utilized, so cell has to shift towards lactose utilization, and for that purpose it needs to produce some extra enzymes called as lactose permease, which is responsible for transporting lactose inside the cell, and beta galactosidase, which is an enzyme which breaks down the lactose into glucose and galactose, and then these two monosaccharides can be utilized. So you will get a stationary plateau after that orange phase, which is a second lag phase where the culture becomes ready for utilization of lactose by producing the mentioned enzymes. Once these enzymes are produced, the cultures can utilize lactose sugar, and by using that lactose sugar, the growth of that culture takes place, and you can see the exponential phase. Then you get a stationary phase there where the rate of formation of new cells is equal to rate of cells which are dying in that flask because of many factors as discussed earlier. And then after the end of stationary phase, you will find that because of accumulation of many toxic molecules, many waste molecules in the flask, changes in the pH in the flask, depletion of the nutrients in that flask, the cultures shows increase in the death rate compared to rate of formation of new cells, which leads to decrease in the total number of cells as you can see from the graph. Since this pattern, that's why this growth curve is called as Diauxic growth curve. So this is observed as mentioned earlier in E. coli, or this can also be observed in the organisms which have operon system or inducible enzymes in them.